Good morning. It's September 16th, and this is your daily brief in science. Here's everything you need to know. The Australian Centre for Disease Preparedness near Geelong is Australia's most secure biosafety Level 4 laboratory, designed to handle the world's deadliest viruses like Ebola and Hendra. The facility employs strict safety protocols, including multiple security checkpoints, airlocks, and protective suits for scientists. Researchers at ACDP wear protective bubble suits and undergo rigorous training to manage risks associated with highly transmissible viruses such as Hendra, dengue, Ebola, and emerging zoonotic threats like salt gully virus and Japanese encephalitis. The center plays a vital role in preventing pandemics through virus isolation, monitoring mutations, and testing vaccines, including work on COVID-19 and other zoonoses. ACDP also conducts animal testing for viruses like bird flu and has contributed to COVID-19 research, using ferrets to understand virus transmission. The article highlights the growing threat of zoonotic viruses driven by climate change, urbanization, and global movement, which facilitate animal-to-human transmission. Research at the center has led to the development of vaccines, such as one for Hendra virus that protects both horses and humans. Scientists study how viral infections begin and spread through the nasal cavity to assess risks and develop outbreak strategies. The facility also researches mosquito-borne viruses, understanding their spread in warming climates and testing interventions, which is crucial with the rise of diseases like dengue and chikungunya. ACDP is equipped with multiple containment layers, including concrete walls and negative pressure laboratories, to prevent the escape of infectious agents. In 2024, the center played a key role in identifying and understanding different strains of avian influenza circulating in Australia. Overall, the article underscores the importance of high-security labs in pandemic prevention and highlights the ongoing challenges posed by emerging infectious diseases in a changing world. Scientists at University College London have made a significant breakthrough in understanding the origins of life. They recreated in the lab how amino acids, the building blocks of proteins, could have spontaneously linked to RNA under conditions similar to early Earth's environment. This involved simulating the emergence of life by linking RNA and amino acids in a simple aqueous setting, akin to ponds or small water bodies on early Earth. The team successfully formed peptide bonds without enzymes or complex catalysts, using reactions that took place in plain water, especially in cold or icy conditions where freezing enhanced amino acylation. They used thioesters derived from pantothene, a sulfur compound present in modern metabolism, which facilitated connections between amino acids like arginine, glycine, and alanine, and RNA strands. The energy from these thioesters, molecules abundant on early Earth, helped drive the chemical reactions, bridging theories of the RNA world and the thioester world. The reactions were spontaneous, simple, and selective, suggesting that early biochemical processes could have occurred in modest environments without harsh conditions. The research highlights the potential role of natural cycles, such as shifts in salinity and temperature, and environments like shallow lakes or coastal areas with freeze-thaw cycles that could have concentrated molecules. While this advances our understanding, questions remain about how RNA developed selectivity for specific amino acids, a key step toward genetic coding. The findings support the idea that early biochemical reactions could have been energy-driven and occurred naturally, setting the stage for the emergence of organized biological systems. This work offers new insights into how proteins and life itself might have originated, not only on Earth but also on other celestial bodies like icy moons such as Europa or Enceladus. A new study presented at the European Association for the Study of Diabetes annual meeting suggests that cannabis use may nearly quadruple the risk of developing diabetes. The research analyzed electronic health records from 54 healthcare organizations, including over 96,000 cannabis users and more than 4 million controls, tracked over 5 years. The findings showed that 2.2% of cannabis users developed diabetes compared to 0.6% of non-users, even after adjusting for various lifestyle and health factors. Researchers propose that the increased risk could be linked to insulin resistance and dietary behaviors often associated with cannabis use. However, They emphasize that more research is needed to clarify the biological mechanisms and causality behind these findings. The study has limitations, including its retrospective design, potential misclassification of cannabis exposure, reliance on self-reported data, and incomplete information on consumption patterns. The authors acknowledge these factors may influence the results, especially considering confounding variables. As cannabis becomes more accessible and socially accepted, the study highlights the importance of metabolic monitoring and public health messaging. Healthcare providers are encouraged to discuss cannabis use with patients to better assess and manage diabetes risk, particularly in regions where legalization is expanding. Maria-Jasin, a leading molecular biologist at Memorial Sloan Kettering and Weill Cornell, has received the 2025 Perlmeister-Greengard Prize for her significant contributions to DNA repair research and gene editing. Her work has advanced our understanding of genome stability and influenced cancer therapies. Jasin's research on homologous recombination has been pivotal in revealing how cells repair DNA and maintain genetic integrity. Her studies on BRCA1 and BRCA2 genes have provided insights into how these tumor suppressors prevent breast cancer by facilitating proper DNA repair. This understanding has led to targeted treatments, including PARP inhibitors, which are used in personalized cancer therapy. She demonstrated that inducing specific DNA breaks can stimulate homologous recombination, laying groundwork for early gene editing techniques. Her current focus is on DNA repair in breast tissue during development and disease, aiming to uncover new vulnerabilities in cancer cells. Her findings have fundamentally changed cancer biology by clarifying the role of homologous recombination as a key repair pathway. Jasin's work has also established principles that underpin modern gene editing technologies like CRISPR. She holds notable positions at Sloan Kettering and Weill Cornell, is a member of the National Academies, and has an academic background from MIT, Zurich, and Stanford. The Perlmeister-Greengard Prize, awarded at Rockefeller University, recognizes her scientific achievements and perseverance in advancing biomedical research. Researchers at the Technical University of Dresden have developed a laser-based drilling system designed to penetrate icy surfaces on extraterrestrial bodies such as comets and Mars polar caps. This technology offers a potential alternative to traditional cryobots, which require significant power and are affected by environmental challenges. Current tests have successfully drilled about 25 centimeters into ice, demonstrating the feasibility of laser drilling for space exploration. To become fully operational, the system needs improvements to extend borehole depth, increase stability, and prevent issues like dust coating the laser mirrors. Laser drilling provides advantages over conventional methods by being more compact, consuming less power, and avoiding physical contact with ice. Traditional cryobots often demand kilowatt-range power supplies and face hurdles like ice sublimation in vacuum environments. The laser creates boreholes approximately 6.15 millimeters wide, sufficient for scientific instruments but requiring adjustments for larger applications. It uses a 1550 nanometer infrared wavelength, which is strongly absorbed by ice, enabling efficient melting with low power. Experimental results show the laser can drill through various ice types at speeds between 1 and 3.1 meters per hour, using just 10 to 20 watts of power. Tests in vacuum chambers confirmed its capability to drill at these speeds, with the fastest reaching 3.1 meters per hour at only 10 watts. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.